today we're going to be discussing molecular biology workflows in Lasergene. To start today's presentation, I'll give you some brief background on DNASTAR as a company, as well as our Lasergene software. And then for the majority of the webinar today, I'm going to be doing a live demonstration of the software. And we're going to touch on some of the most common molecular biology workflows that our customers use our software for. These include cloning and primer design projects, Sanger sequence assembly and alignment, as well as multiple sequence alignment and phylogenetic tree generation. DNASTAR has been in business for over 30 years, and we've come a long way during that time. Founded in the lab of Dr. Fred Blattner at the University of Wisconsin, DNASTAR's commercial efforts were originally focused on DNA sequence analysis and molecular biology software. As the fields of molecular biology, genomics, and biochemistry evolved, so have we, providing tools for next-gen sequence analysis and most recently for protein structure analysis and protein structure prediction. In addition, all of our software is available on a desktop platform as well as on the DNASTAR cloud. This long history and the wide use of our software applications has made DNASTAR the leader in citations in peer-reviewed journals. For all of the last 30 years, we've been cited more than any of our competitors combined. And in fact, just earlier this year, we surpassed 50,000 total publications in peer-reviewed journals. DNASTAR is a global software company. We're headquartered in Madison, Wisconsin, and we have the full support team and technical team in-house to address any issues you may encounter. And perhaps more importantly, we also use the feedback from our customers to make our software even better. In addition to our in-house support team, we have distributors around the globe to supplement that world-class support and customer feedback. So our software supports a wide variety of applications. Today we're going to be talking about Lasergene for molecular biology workflows, and I have a few images from those workflows shown here. The molecular biology suite includes tools for cloning and primer design, Sanger sequence assembly and analysis, SNP analysis for those projects, as well as multiple sequence alignment and phylogenetic tree projects. So we'll actually be doing a live demonstration of these tools, but before we get into that, I just want to discuss some of the other software tools I mentioned. The Lasergene Genomics Suite provides tools for next-gen sequence assembly and analysis, including variant analysis as well as gene expression analysis. Our Structural Biology Suite includes tools for protein structure, prediction, and analysis. We have other webinars talking about some of these other software applications. I just want to mention them briefly today. And then finally, point out that ease of use and flexibility of access is a hallmark of all of the software I've discussed so far. All of the Lasergene software is available on desktop platforms as well as cloud platforms that allow you to access your data and your projects from anywhere in the world. And in addition, we have flexible licensing options, so we can really provide you with the best solution for your organization's needs. So with that, I'm going to now get into the software demonstration. And right now, you should be seeing my Windows desktop. And here I have open the DNASTAR Navigator. And the DNASTAR Navigator is a starting point to accessing all Lasergene software. And you can see here the software is grouped by workflow. Again, you notice the molecular biology, next-gen sequencing, and structural biology components we talked about, as well as the DNASTAR cloud. Today we're going to be focused on these molecular biology tools. And you'll notice as I hover over each item, I get a short description of the application, as well as links to videos, written tutorials, as well as sample data. So you can see there's a lot of resources here for those of you who are just getting started with Lasergene software. So as I mentioned, one of the first and probably most common workflows with our software in terms of molecular biology workflows is cloning and primer design. And so that's where we're going to start our presentation today. So for that, I'm going to click Seq Builder. So Seq Builder can be used for a variety of sequence editing and annotation projects, as well as virtual cloning and primer design, which we'll look at today. So to start, I'm going to create a new cloning project. And many customers have large cloning and primer design projects that they're working with, and Seq Builder provides a great platform for organizing and maintaining all of your sequence data in one place. So just as an example here with our new cloning project, you can see I already have some folders set up, so I could just start bringing in my sequence data, for example, into the Inserts folder. I can also create new folders here if I want to organize my sequence files in a different manner. So we'll just jump right in, and I'm going to show you first a simple restriction enzyme cloning project. So to start, I need to bring in the sequence I want to clone. And to do that, I'll select Import Sequences to Project. And then I'm going to select the sequence I want to use. In this case, I'm opening up a Seq Builder document, but you can bring in any GenBank or FASTA formatted file in addition to any Lasergene formatted file. And so then you see the sequence is added to the Inserts folder. I can double click to open it up and look at the sequence. Here we have the sequence opened in the linear map, and you can see kind of a high-level view of the sequence, all of the annotations along the sequence, as well as the enzymes that cut the sequence. So now that I have my insert sequence added, I need to bring in a vector. For this example, I'm going to bring in a vector from the DNASTAR vector catalog. And this catalog is updated periodically, and it's installed with every Lasergene license, or excuse me, every Lasergene installation. So you'll have access to this vector catalog when you start doing your cloning projects. In this case, I'm going to bring in a restriction vector. So I'll just select the restriction vector I want to use from the list. And I can copy this sequence, or I find what's easiest is to just drag that into my vector catalog, or to my vector folder, rather. And so then that keeps a copy of the vector in the cloning vectors project, as well as adds the sequence to my cloning project. So I'm finished with the vectors catalog, so I'm going to go ahead and close that. And to look at the vector sequence, I can double-click the file name from the vector folder. And you see here I have the circular map of my vector. So now I have my insert sequence and my vector. I'm ready to begin the virtual cloning project. So for restriction cloning, we're going to select enzymes that cut on either side of the portion of sequence we want to clone. In this case, I want to clone this entire TN5 transposon feature. So if I select that, I can see at the top of the window the range of sequence that's selected. So I want to make sure I select enzymes that cut close to this range and include the entire range. So visually, I can see where those enzymes are. Before I select enzymes, I just want to point out really quickly, I have an enzyme filter applied here to only show enzymes that cut at unique sites, so only cut once on my sequence. You'll see there's a variety of other filters you can apply. You can also select specific enzymes. If you have a set of enzymes that you're working with, you can create a filter for just those, or you can search for an enzyme from the alphabetical list. So let me go ahead and make this a little bigger for us. So for this example, to get this entire range of sequence, I'm going to select the ACC651 enzyme, and then I'm going to shift-click the APA1 enzyme. So you can see that selects that entire TN5 transposon feature. And you'll notice at the top of the window, I see the range of sequence selected, and I can just verify that I did select those two enzymes. And now to create the insert, I'm just going to say copy restriction insert. And then to clone it into my vector, I'm just going to select those same two enzymes. I'm going to make sure we single click and shift click. And so just to kind of compare, you can see the two selection feedback items show the same range, or excuse me, the same two enzymes selected. So now all I have to do is go to cloning, clone restriction insert. And that's going to bring up the clone insert dialog. So this is sort of the last step before creating the virtual clone. And this is just allowing us to see whether the ends are compatible. In this case, they are. You'll know if they aren't compatible because these two checkboxes will be empty. And in that case, you may need to simulate adjusting the sequence ends by using the fill-in or trim blunt options, or by dragging the arrows manually to adjust the sequence ends. But in this case, everything looks good, so we can go ahead and click clone. And so then we get a circular map of our insert cloned into the vector. And back to our project window, we now see we have the clone added to the clones folder. So we can name this clone. You can name it actually in the last screen I showed you as well. You can name it here, and then you can save your project and keep adding inserts and vectors. Whatever data you're using in your lab, you can add that all to one project if you want to keep it organized that way. So that's a simple restriction enzyme cloning project. I'm going to show you one more cloning workflow. I'll go ahead and close out of all of this. So in this example, I want to show you how to first design PCR primers and then simulate amplifying the PCR product and cloning it using TA cloning. So I'm just going to open another sequence file. And in this example, I want to clone this gene, this kinase gene here. And so again, notice you have all of your GenBank annotation information here. There's just one gene present in this sequence. So before we were cloning a large portion of the sequence, now we're just going to clone this one gene. And so the first thing we need to do is select the region of interest. And now I'm going to design PCR primers. So to do that, just click Priming, Create Primer Pairs. And here we have some options to specify primer characteristics as well as experimental conditions. You can see some of those options down below. You can also choose to create primers within a range of your selection. But in this case, I want to create primers exactly at the selection, and I'm going to leave all of the settings at their defaults and click OK. So really, with a single click then, SeqBuilder has created located primers with optimal conditions for those settings. And at the top of the window, you'll see the primer view. So here you see our sequence as well as the top strand primer that was created. You can quickly toggle over to the bottom strand primer to look at that as well. And then down below, we have some feedback about our primer, including any dimers as well as hairpins that are found. And SeqBuilder will show a warning here if there's any issues with these primers that would cause either mispriming or poor primer performance. In that case, you may want to edit the primer, and you can do that really easily in SeqBuilder just by, for example, if you want to lengthen the primer, you can just drag the arrows on either end. In this case, I want to keep the default primers because we really did get good quality primers with the default options. You can also introduce restriction sites for restriction cloning or codon changes. Again, just if you want to avoid any dimers or hairpin problems. So I have my PCR primers, and now I want to simulate amplification of the PCR product. So to do that, I'm just going to go back to the priming menu and select Create Insert by PCR. And now again, we see we have a new cloning project, and SeqBuilder automatically created, excuse me, I'm having issues with my full screen here. So SeqBuilder automatically creates an insert sequence from the PCR amplified insert. So we can double click to open that up and see what that looks like. So here you see we have our gene as well as the 3'A overhang, which is simulating the TAC amplification process. So then, to do the actual cloning project, all we need to do now is go to cloning and select our cloning method. So you see we have some other automated cloning methods here, including topo cloning and gateway cloning. Here we're going to do a TA cloning project. So I'll select TA Cloning Advisor, and you'll notice my insert is already selected for me. If I had multiple inserts in my project, I could choose another one from this dropdown. And then here I just need to select the vector I want to clone into. So go ahead and select a vector, and then click Clone. And then SeqBuilder automatically creates clones in the forward and reverse orientation. So again, here we've kind of built a cloning project from scratch. You can continue adding to this with other virtual clones for your projects. So that's just 2 cloning workflows. A lot of the other virtual cloning methods work very similarly. At this point, though, I'd like to move out of cloning and talk about how to assemble and analyze sequencing data that you may have.
Katie, before you do that, thank you. We've had quite a few questions come in. I'm just going to get to a few of them, and others will come back to possibly after the webinar directly with some folks. The first one, right in the cloning, the question is, can I use my own vectors for cloning projects?
Yes, absolutely. So I just showed how to grab a vector from the vector catalog that's installed with Lasergene. But you can definitely bring in your own vectors, and you can bring those right into your cloning projects, and those will be saved for future use. So for example, if you have vectors that you're working with all the time, definitely recommend bringing those into projects and saving them so they're easily accessible. You can also add your vectors to that vector catalog I showed you. So depending on how you want to use the vectors, there's a couple different options, but either way you go, it's very easy to bring in your own vectors.
Okay. And actually, the next question relates to the screen that you have up here now, the navigator screen. We see near the top something called Primer Select. And the question is, why are we doing primer design in this other application, SeqBuilder? Is there some use for this Primer Select that's outside of what's available there?
Yeah, that's a great question. So SeqBuilder is mainly focused on PCR primer design. And as you saw, there were just kind of a few options just designed to make the PCR design, or excuse me, the primer design as simple as possible. Primer Select is used for more general primer and probe design. And additionally, Primer Select has a lot more advanced options. So if you're really heavy into primers and you need to be able to access more of the advanced options, you can go into Primer Select and design your primers there. And then you can actually build primer catalogs. So any primers that you design in Primer Select, you can use those in SeqBuilder. So the idea is just to make it as streamlined and simple as possible. But for those that need the advanced tools, we provide that as well. And we also have capabilities to create sequencing primers, which I'll get into a little later.
Okay, I think that's all the questions we'll take for now. We'll let you keep going into the other parts of the webinar. But,
okay, great. Thank you, Tom.
And I would encourage, though, the participants to please continue to chat the questions in. They're great, and keep them coming.
Yes, agreed. Thank you. So as I mentioned, the next set of workflows I want to show you all pertain to handling Sanger sequencing data. This is another very common use of our software. When I mentioned the number of citations we have, many citations still use, many researchers are still using Sanger sequencing data for their projects and for their work. And SeqMan Pro provides a lot of really nice, easy-to-use tools for handling that data as well as doing downstream analysis. So I'm going to show a couple of workflows in SeqMan Pro. The first workflow I want to show you is for this mumps sequencing data. So this is a Sanger sequencing project for the mumps virus, and I have a folder of trace files here. And all I need to do is just drag this folder onto SeqMan Pro, and that's going to load up all those sequences. And then from here, you can actually just click Assemble, and SeqMan Pro will do some end trimming based on sequence quality and start to build contigs. And that all happens automatically and pretty seamlessly. But just to kind of illustrate the process, I'm going to do the trimming before we do the assembly. So I'm going to just scroll up to the top. I've got 300 trace files here, and we'll just look at one. So you can look at the trace files just by double-clicking, and then you can open your chromatogram, and you can just kind of see the trace along the sequence. And you'll notice, we have some kind of poor quality data at the ends. That's pretty normal. You may also have vector sequence here. And so to really get the best assembly possible, we want to trim out any of that noise before assembly. So you can do that manually. I'm going to just do that quickly here by selecting all. And then I'm going to select a vector. You can just type your vector in, or the first few letters, or you can select one from the drop-down. And then I want to show you just a little bit of detail on the end trimming. So by default, SeqMan Pro will trim kind of at this middle of the road, medium quality stringency, but I'm going to up this to high. And then I'll say scan all, and that's going to do the quality trimming. So that just did the quality trimming there. We did set the vector, so I'm going to go ahead and scan again to scan for that vector sequence. So now you see the sequence ends have been trimmed. You can confirm that by looking at these limits. So you see here the sequence starts at 39, so we have 38 bases trimmed off one end. And you can see if the vector was found because you see the check mark next to the vector name. So if I'd just done the quality trimming like I did earlier, you notice these were all question marks, which meant it hadn't scanned for vector yet. And upside down question mark just means we searched for the vector and didn't find it. So then let's look at that top sequence again. So this is the same chromatogram we looked at before, and you can see that low quality data was trimmed, and as well SeqMan Pro detected vector sequence there and trimmed that away. So these trimming results are usually pretty reliable. At this point, you can go ahead and assemble. But I just want to point out, if you ever want to, you can adjust the end trimming manually just by dragging this arrow. You kind of see the part that's trimmed away is shown in gray. And you can very easily restore that by dragging the bar back. So at this point, our sequences are trimmed for quality and scanned for vector. I'm going to go ahead and assemble them. So with a click of a button, SeqMan Pro is then going to take all of those trace files and start to build a config. And you see that happened pretty fast. And in this case, all of the sequences went into a single config. 291 of the 300 sequences went into the config. So SeqMan Pro will also deal with if there are any sequences that are too short or just not high enough quality, SeqMan Pro won't use those for the assembly. So to understand the results better, we can first double-click the config just to look at an alignment view. And so here you can see, we can scroll along the config to see the sequence alignment. And another option I want to point out is, if you see areas like this area here where we have a large gap and also some conflicts, you can investigate that further simply by expanding the sequence data. to see those chromatograms. So again, you have really full visibility and access to the original data here. So here we can see, for example, in this sequence, maybe we have some low quality data. We can trim that. You also see some gapping in the consensus. We can select that, and that will select the region in all of the reads as well. So if you determine, for example, that a gap is, artificial or, just an artifact of, maybe some poor sequencing data, you can edit your consensus directly by just selecting the portion of the consensus you want to change, and then either typing in a letter, or in this case, I'm just going to delete that gap. So the software makes it really easy to do these micro edits so that you can, get the best consensus sequence for your downstream analysis, or perhaps your, sequence annotation projects. In addition to looking at the contig or the assembly results on kind of this micro level, you can also look at a higher level. So for that, I'm going to look at the strategy view. And this shows me a graph of coverage along the contig. So you can see we have some valleys and peaks with areas of lower and higher coverage. And this is all integrated with the alignment view. So if you want to investigate further, say we want to look at this high coverage area, we can double click. And that just jumps right to the alignment. And we can see, generally it's pretty good quality here. So there's nothing, we don't need to do any, further work there. If you saw a larger peak in coverage, you might suspect some, perhaps a repeat region or something that caused the sequences to align incorrectly. But that looks okay. You may also want to look at an area of lower coverage, and here we see, in fact, the coverage is pretty thin. We've got about 3 sequences here. So the next step you might want to take is to improve some of the sequencing coverage. So to do that, what we can do, and I alluded to this a few months ago, is we can design sequencing primers to improve coverage. So to do that, I'm just going to go to Config, Primer Walk. And SeqMan Pro provides a few options for primer walking. You can look for primers that walk into gaps. That doesn't really apply here, but what that means is, let's say our sequences had assembled into multiple contigs and we wanted to join those contigs together with more sequencing data. We could walk into gaps in that case. Here we just want to improve our sequencing coverage. So I'll select Improve Coverage, and then I'm going to leave the default options, but note that you can specify exactly how deep you want your sequencing coverage to be. And that's going to tell SeqMan Pro where on the existing contig do I need to look for primers to get more sequencing data. So I'll click OK, and you'll see that search happens pretty quickly. SeqMan Pro locates primers along the contig, and those are, I don't know if you can see, let me zoom in. Those primers are also represented along the contig, so you can see the forward and reverse primers. And then you can export this primer catalog, as I mentioned, if you want to actually use those sequencing primers for downstream projects to fill in with more sequencing data. And then you could add that data back into SeqMan Pro for reassembly. So that's kind of a short de novo project. I'm going to show you one more workflow in SeqMan Pro, so I'm going to go ahead and close this. And create a new project. And before I use drag and drop, but now I'm going to add sequences. You can add your sequence data however you like. And then I'm going to select some of the Lasergene demo data. And in this case, again, I have some trace files. But here you'll notice I have forward and reverse reads for 3 samples. So one thing you may want to do, in the last example, we assembled all of our data together as one sample, one project. But let's say you're interested in comparing these different samples. Perhaps you have sequencing data from multiple organisms or multiple conditions, and you want to assemble data from each sample independently for downstream comparison. So to do that, we're going to use the Assemble in Groups capability. But before I do that, I'm also going to do this example as a reference-guided assembly. So I brought in a reference sequence. For this, this .seq is a Lasergene format, but you can bring in any GenBank or FASTA file. You can also bring in multiple reference files. But for this example, I want to assemble each sample against the same reference. So I'm going to select the reference sequence and click Mark Ref. And then, as I mentioned, just click Assemble in Groups. And so then the last step here is just to select the unique part of the sample name. And then I can confirm that I have 3 samples here, and the forward and reverse reads are each going to go into a single group per sample. And then I just click Assemble. And then as expected, we get 3 contigs, 1 for each sample. Again, you can look at this data, the alignment data, by double-clicking the contig. And now you'll see, in addition to the forward and reverse reads, again, we can look at the trace data. We also have a reference sequence. And if you expand the reference sequence, then you'll see if you did a GenBank or Lasergene formatted file, you can see any annotations along that reference. And just as before, you can scroll along the contig and do any kind of micro editing that you need to do to improve your consensus sequence. So then at that point, because it's the multi-sample assembly, perhaps you're interested in then comparing these samples. So you could export a FASTA file with all of your consensus sequences and then say, align them and build a phylogenetic tree. Another thing you can do with any reference-guided assembly, multi-sample or not, is SNP analysis. So for that, I'm going to open up the SNP report. And here you see a pretty detailed report of all the SNPs found in the project. And there's not a lot found here. These are fairly short sequences, and we just have 3 samples. But you can get an idea of the kind of information that you get here. You see the reference base as well as the consensus base. You can see, if your SNP is located in a coding feature or how close it is to a coding feature, the depth of coverage of the SNP, and you can use all of this data for filtering. So say you're only interested in coding changes, you can filter for that right here in SeqMan Pro, and then start to look at those SNPs in more detail. So for example, here we have a coding change SNP. I can select that and then just double click to go right to it. And then you can look at your trace files to sort of corroborate whether that is a true SNP or some kind of sequencing error. So that's just a really high-level overview of SeqMan Pro. It kind of combined de novo, single sample, and reference-guided multiple sample. But you can do different combinations of the 2 workflows depending on your needs. So at this point, I'm going to talk about our final workflow, which is multi-sequence alignments and phylogenetic trees.
Katie, before we leave SeqMan Pro, we've had some good questions come in, and I can share a few of them. First one is about quality scores. Can I view my quality scores in SeqMan Pro?
Yeah, that's a great question. So for these 2 examples, I was really focused on trace data and looking at the chromatograms just visually. But yes, you can definitely supplement your analysis by displaying quality scores. So that tool is right here on the alignment view toolbar. So here we can see quality scores that are derived from the trace data. And this is also a really useful tool. Let's say you don't have trace data if you're just working from FASTA files. If you have quality data associated with those FASTA files, SeqMan Pro will bring that in. And that's a really important tool then for doing some of the editing I showed you, so you can actually see the quality of the base call.
And that actually leads to another question, which has to do with next-gen sequencing data. Does SeqMan Pro handle the assembly of that data?
Yeah, that's a great question. So SeqMan Pro is really designed for, in terms of assembly and alignment, handling Sanger data. But we do have a full suite of tools, including SeqMan Pro, for assembling and then analyzing next-gen sequencing data. And that's part of our Lasergene genomics suite. And for the assembly portion, we have a product called SeqMan Engine, and that's able to handle a wide variety of, different sequencing technologies, including Illumina and Ion Torrent, as well as 454 and PacBio, as well as a whole variety of sequencing workflows. So for these examples, I just kind of showed some simple assembly workflows, but with our next-gen assemblies, we have a whole host of workflows where you can do whole genome assemblies for organisms of different sizes, or exome assemblies, or gene panels. So because there's so many more workflows there, we have a separate application to get those assemblies set up. But then you can do some of the same downstream analysis I showed you in SeqMan Pro with the same tools we looked at today, as well as further options for example, SNP analysis. We have some really nice capabilities for advanced SNP analysis, particularly multi-sample SNP analysis. So it really builds on the workflows I showed you today, but we have separate tools to address the unique needs of our next-gen customers.
Okay, thanks, Katie.
Thank you. So, as I mentioned, we're going to now look at some multiple sequence alignment projects, and for that I'm going to use Megalign Pro. So Megalign Pro is designed to make it really easy to align multiple sequences, and those can be DNA, RNA, or protein sequences. We also provide a variety of alignment methods. And additionally, we have the capability to align entire genomes here in Megalign Pro. So this is often kind of a last step in people's analysis, sort of looking at, they've done their cloning, they've got their sequence data, and now they want to compare, say different consensus sequences between organisms or between samples, and just start to look at, what are the evolutionary relationships between the different sequences, either genes or genomes that they're working with. So to start out, let's just start simple. I'm going to do a basic sequence alignment. So click the Add Sequences button. And again, I'm going to be working out of our demo data folder. And here I'm going to select 5 alcohol dehydrogenase protein sequences from different organisms. So I'll add those. And here you can see, by default, these are all added as unaligned sequences. And they're represented at the top of the view as just gray blocks, meaning no portions of the sequences have been aligned yet. At the bottom of the view, we see the actual sequence data for each sequence. And we can expand that to see the GenBank annotations. So you may be noticing a theme here. I just always like to point out that you really have access to the full sequence file here. It's not just the amino acid residues or the DNA base pairs. It's a sequence as well as any annotations. So what's useful here in Megalign Pro, in this case, I just have the protein sequences. But let's say you're aligning a larger segment of a genome or a coding sequence. You can actually see the annotations to see, okay, where are the genes that I'm interested in, or where are the exons as they compare to, say, each of the other sequences. So I have my sequences added, and to align, it's pretty much one click. You can just click this Run button here. And you'll notice by default, if I hover over that button, it's going to align using the muscle algorithm. But if you want to use a different algorithm, you just click this arrow to the right and select the alignment algorithm of choice. So here I'm going to align using MAST. And you'll see that will take just a few minutes, and Megaline Pro is going to align and gap all of my sequences, as well as build a distance matrix and a phylogenetic tree. So that alignment will finish up here, and we'll look at each of those. So now you'll notice at the top of the screen, my sequences are now kind of represented as a mix of gray and green blocks. So the green blocks are portions that align to the rest of the sequences, and the gray blocks are portions that don't align. And so you can use this view to do kind of view finding and look at, okay, you know, where's this region where we have some poor alignment? And then you can look at in-depth detail of the actual sequence data in those sections. Another downstream analysis option is the distance matrix. So here you can see pairwise distances between each pair of sequences in the alignment. And then if I select those distances, those two sequences are selected in the overview. Another option is the phylogenetic tree. So here you can see a phylogenetic tree that was created during alignment with all 5 sequences. You can then export this tree as is, or you can export it for further refinement and analysis in other phylogenetic tree programs. So that's kind of the most basic alignment option. Another alignment option in Megaline Pro is the ability to group sequences together for an alignment. So let's say, for example, you have sequence data for exons from a single gene in multiple organisms. And you wouldn't want to just align all of those exons to one another, but rather you'd want to group the exons by organism and align the entire coding sequence from one organism to another. So I'm going to demonstrate that exact scenario. In this case, instead of adding sequences in bulk, I'm going to add sequences end to end. And what this allows me to do is define groups for my sequences. So I'm first going to add exons for the RP1 gene from a single organism. And in this case, I'll choose chimp. Again, I'm working from the Megaline Pro demo data. So if you want to go back and try any of these examples after the webinar, you're certainly welcome to. So I'm going to change the name here. You can leave the default name or you can type in a custom name. Here I'm just going to call this chimp. And you'll notice add another is checked. So I have my 4 chimp exons added, and then I'm going to click OK to add my next group. So now I'll add the cow RP1 exons. And notice I only have 3 in this case, but that's okay. That's not going to cause any issues for our alignment. And I can name that cow. And to start, I'll just align these 2 sets of exons. I'm going to uncheck add another and then click OK. So here you can see each sequence added to the row corresponding to the group. So we have our 4 chimp exons and 3 cow exons. And here I'll align using cluster omega. And then you can see how the exons align to one another. So here, for example, if we expand, we can see exactly which exons we're looking at. So here we see the chimp RP1 exon aligned to the cow RP1 exon. And as we scroll along, we can see the different components of our alignment. So that's 2 organisms. If we want to add another organism, we can just click Add Sequences End to End again. And let's say we want to go ahead and add the human exons to the alignment. We can do that. Just call that human and click OK. And then now you see humans added to the unaligned sequences, and we've maintained the alignment of the 2 RP1 exon groups. And then I'm just going to click this button to merge the unaligned sequences into my existing alignment. And we'll continue using Cluster Omega. You could use a different alignment method. If you ever need guidance on what alignment method to choose, we do have integrated help here. We'll go ahead and close that. But then you can see the other alignment options available. You can also do multiple iterations to refine the alignment. But in this case, I just want to do a quick addition, so I'll click Align. And then that's going to incorporate the new group into the alignment. And so again, you can kind of see how that new sequence then aligns to the existing. And as you start to build this alignment, in this case I did it iteratively, but you could do all of the organisms at one time. You may want to look at another phylogenetic tree, and as expected, we see chimp and human closely related and cow more distantly related. And this, is as expected, and we can keep adding sequences to get, a little bit more interesting looking tree. But in the interest of time, I just wanted to do those 3 for you. And then the last alignment method I want to show you, I actually have a completed alignment here showing a MAV alignment. So we've really been focused on gene sequence alignments, but Megalign Pro is a very powerful application and actually allows you to align entire genomes. So in this case, I have an example where I've aligned 3 H. pylori genomes. And this is what the completed alignment looks like. And so you'll notice the sequencing blocks are displayed a little bit differently in this case. They're color-coded by segment, and they're also connected by lines. So you can see, for example, this pink segment is the same in all 3 genomes, and it aligns pretty closely along the genome. But you'll notice in other cases, you have these lines crossing, and that indicates some genome rearrangement. So, for example, some of these shorter sections towards the end of the sequences are rearranged. You notice there's a fairly large rearrangement here, line cutting over. In addition to the genome rearrangements, you'll also notice there's some inversions, and those are denoted by, say, for example, this purple block. It's shown in the forward orientation on the top genome, and then on the second 2 genomes, it's shown below the solid line here, which indicates it's in the reverse orientation compared to this top genome. So here we have a genome inversion. And if we want to look at that in more detail, all we do is double-click on one of the purple blocks, and then that actually is going to kind of rearrange the rest of the blocks to put all of these the sequences in this section all in the same orientation so that we can compare them. And so then, just as we did before, we can look at the actual sequence alignment for that region of the genome. So this is a really powerful tool if you're sequencing, say, microbial genomes, and you want to look at, what are the major differences between the genomes I've been working with. So that actually concludes the alignment methods I wanted to show you. We've just kind of scratched the surface on some common molecular biology workflows. We only have an hour today, so I'm going to wrap up now, but we have a variety of other workflows available.
Yeah, and Katie, we have just a couple questions here, if we have just time to get to a few of them. One of them,
definitely.
what do the numbers at the nodes of the phylogenetic tree represent?
Oh, sure. So those are distances, calculated distances between the sequences. So that's based on the sequence similarity. And those are, I guess, technically they're branch lengths, but they represent the distances between the sequences or the similarity between the different sequences in the tree.
Great. And there was one other question I'd like to get to. It had to do with your earlier example where you were adding in some sequences to an already existing alignment. And I think the question was, can you delete sequences? And if so, what happens then?
Yeah, that's a great question. So to delete sequences, it's pretty similar. Rather than adding a sequence in, you can just right-click and select Remove Sequences. And just as when we added a sequence, it had to add it into the alignment and kind of run through the alignment to incorporate the new sequence. Megaline Pro will go through a similar process when you remove a sequence to remove any gaps that may have been introduced by that particular sequence. So that way you won't get leftover gaps in your alignment that were caused by a sequence that's no longer in the alignment.
Okay, thank you, Katie.
Thank you. So as I was kind of alluding to, we just did a couple workflows and a few applications. We have a variety of other workflows available, even just in molecular biology, not to mention genomics and structural biology. If you're interested in learning more about some of the topics we discussed today, we do have an archive of webinars much like this one that talk about some different workflows. So you can visit our webinars page to see some of those. We also have a wealth of videos, 2 to 3 minute videos. If you don't feel like investing in another entire hour, we have some short videos to show you different workflows. Those are great for getting started with the software. They're also great for getting some more in-depth information about a particular workflow. And then finally, I just want to mention we have another webinar coming up hosted by my colleague, Matt Kaiser. Matt's going to talk about our next-gen sequence alignment and analysis tools, and specifically discussing accuracy in those next-gen sequence alignments and variant analysis projects. So that's going to be a really great webinar, and I certainly encourage anyone who may be on the call today that is interested in next-gen sequencing to attend that webinar as well. As for today's webinar, if you missed any portion of the presentation, we'll be posting a recording of that here on the archived webinars page, and in addition, we'll email you all a link to that recording as well. So that's all for today. Thank you all for joining us. We will follow up with anyone whose questions we did not get to, and otherwise, we hope to see you again soon.